Well, today we're going to be talking about something called circular causalities. It's also called causal circularities, maybe better known as the chicken and egg scenario, right? Which came first? There's a whole bunch of these in biology, especially biochemical pathways. And I thought we'd get started with a few of the more well-known ones, spend some time discussing those, and then I'm sure some of you can come up with your own. I didn't cover all of them. There's a lot of them. So we'll keep this pretty fluid. Feel free to interact at any point in the presentation. So there's many times in nature where you have some sort of substance, A, that is turned into product B, but B is also needed to produce A. And that's where there's a causality here. A is needed to cause B, but B is needed to cause A. And so how can you get those sequentially? How would you evolve one of those without the other one? They simultaneously have to be in existence in the same cell in order for life to even get started. And sometimes there's even a three-way factor of this. So take a look at DNA, protein, and RNA. Well, DNA contains the genes that code for proteins, but some of those proteins are needed to synthesize DNA, not only to synthesize the building blocks of DNA. There's a whole bunch of enzymes needed to create, the adenine, guanine, cytosine, thymine, plus the ribose or deoxyribose sugar that goes on there. In fact, at first it's made as RNA and then converted into DNA bases through a special enzyme. And then you need another enzyme to add the phosphate groups on there to make the nucleotides. And then another protein is needed to link up those building blocks as nucleotides to make long chains of DNA. You also need DNA to serve as a template to make RNA, like messenger RNA, which of course involves proteins like RNA polymerase involved in transcription, where you make if DNA is sort of the coded information that you would find on a hard drive, like the zeros and ones, the RNA would be like a printout. It's a different language, but it's coded by what's on the hard drive. But the printer itself would be the protein, the RNA polymerase. And in the case of some viruses, RNA viruses like HIV, they have an RNA genome that has to be turned back into DNA so the host cell can unwittingly use that viral DNA and make viral proteins and a whole new virus. So there's an enzyme called reverse transcriptase that will turn RNA into DNA. So which came first? We have these proteins that serve a special role of speeding up chemical reactions called enzymes, and they can speed up reactions by millions of times faster than they would otherwise happen. Especially if it uses energy like ATP to power that reaction, it can overcome energetically unfavorable reactions and make them happen. So for every substrate, and that substrate could be as simple as CO2, as complex as DNA, it could be another protein, it could be a small molecule. There are enzymes that will take that substrate and turn it into a product. But it's crucial that there's also regulation of this. Since these things are so fast, you don't want to have enzymes active all the time. Sometimes they're even pre-assembled in an inactive form. Those are called zymogens. An example are the proteases, the protein chopping enzymes in your stomach, pepsin and trypsin. They're first synthesized as sort of a corked enzyme that's inactive, called pepsinogen and chymotrypsin. And when you eat food, there's a complex series of signaling that goes on that tells specialized cells in your stomach to start producing hydrochloric acid and basically increase the proton concentration in your stomach. This helps lower the pH from, neutral pH of 7. This goes orders of magnitude down, even down to pH of 1. So just going from pH of 7 to 6 is a tenfold increase in the hydrogen ion concentration or proton concentration. It's more acidic. So yeah, at pH of 1, you could dissolve metal. And when that pH drops, it activates these digestive enzymes, so they start chopping up proteins. You wouldn't want them on all the time. It could, especially if you didn't have a protective mucopolysaccharide layer on your stomach, you could start digesting your own stomach, and that's what happens with ulcers, typically from a bacteria called Helicobacter pylori, which can turn ammonia into, it basically turns your stomach into more of an alkaline localized environment that offsets the acid, and it eats away some of that protective slimy layer in your stomach, eats up some of those polysaccharides, and then the acid and the proteases can get in there and start eating holes in your stomach. There's a lot of enzymes that are paradoxical in the sense that, let's say in bacterial cell division, the cell wall needs to weaken where it's going to pinch off. So you have the bacteria has enzymes that weaken that cell wall, digest it, and then there's other proteins that will pinch the cell membrane underneath tight. It'll divide in two, and then you got to regrow the membrane and the cell wall around it. Now, what if that cell evolved the degrading enzymes first before the building enzymes? Yeah, you got death. You got a disaster on your hands. You've got to be able to regulate these enzymes, not only how much of them you make, but whether they're turned on or turned off. And there's even some secondary molecules like inhibitors that can bind either to the active side of the enzyme. So sometimes those inhibitors look just like the substrate, but they don't get turned into a product, and they will occupy a little pocket on the enzyme that normally binds to a substrate and turns it into something else. And this one's shaped like it, but competes for that site. You have a competitive inhibitor, so now the real substrate can't get in there. And oftentimes, at the end of this long biochemical pathway, the last product on there acts as the inhibitor for the first enzyme. Not the second, not the third, but exactly where you would want the master regulatory enzyme at the beginning of the pathway. So you don't end up with a buildup of all these intermediates or quickly turning all your precious resources into product, and you don't need all that product. Or getting a backup, a buildup in the assembly line if the enzyme isn't turned back on fast enough. There's also inhibitors that will bind to an enzyme on another site called an allosteric regulator. So this allosteric pocket will change the shape of the enzyme active site so it can no longer bind to its substrate, and that's another type of regulation. And you can just digest the messenger RNA that codes for that enzyme so that you stop making more of that enzyme. So there's multiple different ways that the cell has to regulate that, and it has to be very carefully choreographed. But there's many types of digestive enzymes that you have to be careful with, like nucleases. Imagine, so these are enzymes that digest nucleic acids like DNA and RNA, and we don't think about it, but we're actually repurposing a lot of the DNA that we eat. We think about carbohydrates, proteins, and lipids or macronutrients, but there's a lot of DNA and RNA that you have to digest as well, and you even reuse some of that. You don't have to build it from scratch every time. It's called the salvage pathway. So you have all these enzymes that chop stuff up, but it can't chop up your body. Like your pancreas produces some of these digestive enzymes. It makes pancreatic lipase, which digests lipids, and it makes a proteases as well. And if you get any sort of pancreatic disease that causes these enzymes to leak out, you get autodigestion of your own pancreas. And that's uncomfortable, yeah. You can even see it on an x-ray sometimes because it'll start digesting the membrane lipids, and basically those will combine with calcium, serum calcium, to form soap scum that shows up on a radiograph. But it's painful. You're digesting the wrong tissues. In fact, if you starve yourself enough so you run out of carbohydrates, you run out of lipids, you start digesting your organs and turning protein into fuel as a last-ditch effort. And of course, all this has to be regulated. The very first cell has to have the proper balance. Why does the first cell need enzymes? Because otherwise the reactions would happen too slowly. The cell would disintegrate long before some of these chemical reactions would ever happen. But if you have an enzyme that is a nuclease that somehow evolved and not the ability to regulate it, well, then all your precious DNA and RNA that cell worked so hard to obtain or evolve is now going to be digested in a matter of minutes. So you have to be able to control that. Here's another classic example in biochemistry where you have thousands of different receptors on the surface of your cell membranes that are embedded in the membranes as proteins. Sometimes they even have some sugar groups attached to them and make them fancier, sort of antenna. And every receptor has to have a ligand that it binds to, some sort of signaling molecule. If one cell wants to communicate with another cell, it'll often release some sort of ligand, or two cells can actually touch each other and there'll be a ligand on the membrane of one cell and a receptor on another, and it'll tell the cell, hey, turn on this gene or turn off that gene. And there's actually quite a few steps that come after the receptor binding by the ligand. Now some ligands activate receptors, some others inhibit the receptor. So we have agonistic, where they activate the receptor, and antagonistic, where they block the receptor activity. So for instance, guys and girls have testosterone and estrogen receptors. There are things in our food that come from plastics, like bisphenol A and phthalates, that can bind to and activate certain estrogen receptors or block them. So some of these can effeminize men or masculinize women, and it doesn't take much to really throw things off, because what happens with that receptor is it causes sort of a mechanical chain reaction through the membrane, and on the inside of the membrane there's a part of that receptor that will kickstart a whole signal transduction cascade. So for instance, it will phosphorylate another protein by adding a phosphate or dephosphorylate or do something that then goes and reacts with a bunch of other molecules and amplifies that signal. Eventually, at the end of that long cascade, there's what are called nuclear transcription factors, and these will go through the nuclear pore. They will bind to DNA and either turn on and turn off genes. Let me give you an example of one, just so you can be marveled at the complexity of this, where you have something binding to a receptor, the receptor gets activated, that starts a whole domino effect of signal transduction, where you amplify the signal in the cytoplasm, eventually resulting in a protein that goes into the nucleus and turns on and turns off a gene, transcriptional activation or deactivation. So this is a very common but complex pathway called the MAP kinase pathway. They have hand-waving terms that they use, but no one's actually mechanically shown. What they might try to say is that there was a gene duplication, and one of those went on to be tinkered, modified, mutated, until it eventually served another purpose. So,
but it would still, it would have to happen extremely quickly.
And the other problem is, until this gets done tinkering and being modified, it's actually an evolutionary disadvantage, because now you're making something that at best doesn't do anything, right, and at worst can actually be a monkey wrench in another pathway. What if it has enough of it retained that it still binds to a receptor but doesn't activate it because it's mutated or something? Now it's occupying a site and acting as an inhibitor. And the cell's wasting resources making something that doesn't do anything. It costs energy and it costs building blocks to make that other item. So evolutionists would say that at some point, especially in these signal transduction molecules, they get repurposed for another function. So what worked in this pathway, never mind how you got that first pathway to begin with, but you take some of the same parts and they can be used as activators for a different pathway that turns on and turns off a different gene. So it's sort of like gene duplication or repurposing the parts that already exist, but it doesn't do a good job of explaining the origin of these pathways. And I want you to see here, this is a endothelial growth factor receptor. So this white part is the membrane. These are phospholipids here that make up the membrane that's fluid, so you'll see them moving around. This blue thing is the receptor, and the red thing is the ligand. So it's going to actually change shape when it binds to this.
And differentiation by binding to EGFR, a transmembrane protein that belongs to the family of receptor tyrosine kinases. When EGF binds to EGFR, it locks the receptor into a specific conformation.
So this is the part outside of the cell, is binding to the substrate, and the membrane receptor it's a transmembrane receptor, so that protein spans the entire membrane, and here's the intracellular side of that receptor.
Allowing a second receptor, tyrosine kinase, to bind effectively. In this animation, a HER2 molecule binds EGFR. In contrast to EGFR, HER2 does not need an extracellular ligand like EGF to be able to dimerize.
So the dimerize means these two come together and work.
To take place on the intracellular.
Did you see how they interact on the inside of the membrane now? Now that they're close enough to do that? That's going to start a whole signal transduction cascade. It's going to amplify the signal.
Intracellular domains of EGFR can recruit the downstream signaling molecules of the MAPK cascade, GRB2 and SOS. GRB2 is effectively the mediator protein for the signal from the receptor to the soluble intracellular proteins. SOS serves as a docking site for RAS. Upon binding of RAS, it is able to exchange the GDP in the cofactor binding pocket with GTP, thereby activating RAS. RAS then dissociates from the activation complex while staying attached to the cell membrane. In this manner, one GRB2 SOS complex can activate multiple membrane-bound RAS proteins, thereby leading to a signal amplification. The signal cascade continues with RAF. In the inactive state, two 1433 proteins are bound to RAF, one of them blocking the interaction surface with RAS. To activate RAF, it first needs to get rid of the N-terminally bound 1433 protein. Separated by 1433, it is possible for RAF to change its shape and bind to RAS. Multiple activated RAS proteins, combined with RAF, form a structure called a nanocluster, which can then recruit downstream signaling proteins. The sole removal of the inhibitor protein 1433 is not enough to activate RAF. Another protein called SRC is needed to phosphorylate RAF to fully activate it. A scaffolding protein binds RAF and SRC and forms a complex. The scaffold increases the local concentration of SRC and RAF, allowing them to interact and ultimately facilitating the activation of RAF by phosphorylation. Activated RAF is able to activate the downstream signaling molecules, MEK and ERK. Once again, scaffold proteins are required to do that efficiently. Activated ERK translocates to the nucleus.
So that's your transcription factor. Now we're inside the nucleus. We're heading there. That was a kinesin molecule that carries vesicles through the cell. If any one of the components here was missing, the whole thing would stop. That's a nuclear pore protein. There's hundreds of proteins involved in
activates MIC, a transcription factor involved in the regulation of 15% of all genes. Normally, MIC is degraded rather quickly in the nucleus by proteasomes. But by activating MIC, the half-life is increased and it can accumulate in the nucleus.
That's like a recycling bin.
MIC can then form a dimer with MAX.
Tree
This
chipper.
dimer can subsequently bind specific motifs on the DNA.
So there's your DNA binding
Aside
protein.
from the DNA, MIC can bind histone acetyltransferases. Acetylating the histones makes them negatively charged. Since DNA itself is negatively charged, this causes dissociation of the histones and allows the transcription machinery to access the DNA and start the expression of genes located there.
You're unraveling the DNA so that it can be read, because normally it's wrapped around proteins called histones.
Another way of influencing the gene expression is by cross-linking two DNA strands via Micmax and Madmax dimers.
Biochemists have fun coming up with these names.
With these complex interactions, the docking of a simple extracellular growth factor can cause a shift of the protein expression pattern of an entire cell.
All right, this is Ribosome Studio. They have some really cool animations. So here we have not only several circular causalities, but we tap into the concept of irreducible complexity, where you have to have all of these components in place in order for the completed pathway. A mutation or a missing component. And I mean, did you guys even keep track of how many proteins were involved in that entire pathway? You should see a schematic diagram. It looks like a map of like New York City or something. There's just so many, and all that's regulated too. This is the simplified version of that pathway. It's happening at an incredibly fast rate, just with like Brownian motion and the molecular kinetic energy. Each one of those blobs you saw was made up of thousands of atoms. These proteins are very complex, usually a couple hundred amino acids. So each one of those somehow has to end up in the cell at the same time. Yeah, and that means you have to have different activators and inhibitors, basically regulatory regions for each set of genes to be able to turn on and turn off. And simple bacteria do this as well. They just don't have a nucleus. But if an E. coli has receptors and hey, glucose is coming from over there, and they have these receptors for glucose that basically tells the flagella to start spinning up and moving, and it starts to move this way. And if it gets further away, it stops, reverses, and tumbles around. And maybe it goes this way. Oh no, we're getting further away because the receptors aren't getting triggered as fast. So then it tumbles around again. Oh, let's go this way. Hey, it's getting stronger, hot, until it gets to the source of the food and gobbles it up. And then you have to have different receptors for taking in the glucose, transporting it across the membrane. And then there's, all the enzymes of glycolysis and the citric acid cycle, electron transport chain, just to be able to utilize that food source.
Well, that was the other thing I wanted to say is that, this is one infinitesimally small part of what's actually going on inside the cell.
A tiny, tiny little snapshot. Yes, so it depends on the pathway, but and you might be able to envision okay, can you cut out some of the middlemen? Possibly, but you can't envision a pathway where there's only two things involved. I mean, there's a minimum complexity to get life going, and it's not just going to be hey, I happen to get this molecule and this molecule, and they bind to each other. Well, then what? We're talking about whole complex systems that are needed for life. So here's another circular causality that we call Eigen's paradox, and it's the observation, back in the early 70s, that we have all these DNA repair mechanisms that are required to maintain the integrity of our genome. That information is really important. It can handle a certain number of mutations, but you need to make faithful copies of DNA with greater than 99.9% accuracy, or the mutations accumulate so fast that the organism basically dies from too many mistakes. It's called error catastrophe. The problem is the very enzymes involved in DNA damage recognition and repair, and a recent paper just came out where they found over 605 proteins involved in recognizing and fixing mistakes in the DNA. Those are themselves encoded by DNA. So which came first, the DNA or the repair enzymes? So Monty and I are part of a team of 5 scientists who meet each week to work on these scripts for animations. We have a talented animator that makes these fun little 10 to 15 minute long clips called long story short. So if you go on YouTube, search long story short and evolution or abiogenesis. And I have a clip here where we talk about Eigen's paradox.
Meet Manfred Eigen. He realized that the information in RNA or DNA that's required to maintain life would experience an error catastrophe without complex enzymes that can recognize and fix this damage to DNA. Without these error correction systems, you would essentially die of old age before you were even born. Fortunately for us, all living things are equipped with complex repair mechanisms. Unfortunately for abiogenesis, these mechanisms need to be encoded within DNA, but the DNA can't exist without the repair mechanisms. This is Eigen's paradox, a chicken and egg problem that has been around without a solution for decades. We can't have long strands of DNA or RNA unless it can repair itself, but it can't repair itself unless you have long strands of DNA.
All right, I remember in my undergraduate microbiology course, we had a lab where we had these strains of E. coli. This particular strain had just one of the DNA damage repair enzymes mutated. So there's at least 3 that are involved in just fixing the damage that occurs by ultraviolet light damage. And so one of them was knocked out or mutated. And we grew these mutants on Petri dishes, and we put half of the Petri dishes on a windowsill and the other half in the drawer where it's dark, no ultraviolet light. You know, in the matter of 3 days, we looked at those Petri dishes, and all the ones in the drawer grew colonies of E. coli. The ones on the windowsill were completely sterile. They had acquired so many mutations in a matter of days, just a few generations of E. coli, that there was too many errors in their genome to survive, and they all died. Yeah, it can't, I don't remember if it couldn't repair thymine dimers or if it was like an excision repair enzyme, but yeah, one of the components that is needed to fix the kind of damage you get when ultraviolet light hits DNA, because DNA, its peak absorbance in this electromagnetic spectrum is in the ultraviolet C range, like 220, 240 nanometers. Yeah, the most damaging form of ultraviolet radiation happens to be the one that the ozone layer also absorbs. So if we didn't have that gaseous sunscreen, which is made from oxygen, it gets turned into O3, O2 gets turned into O3, our Earth would be sterilized so fast. We would have horrible cancer rates. You guys remember years ago when before they banned chlorofluorocarbons and aerosol sprays and like Freon or air conditioners, and they would leak out of old refrigerators that were thrown in the dump, that we had this hole in the ozone layer. There was a bad one over Australia where like sheep would get horrible skin burns and skin cancers from being under this hole in the ozone layer, and they banned these chlorofluorocarbons that happen to destroy ozone, and it's sort of healed itself since then. But it's another one of those paradoxes. It's like most of the oxygen on Earth comes from photosynthetic organisms, but you need the ozone in order to protect those photosynthetic organisms. It's like the primordial Earth would be such a hostile environment. We can't even create a single-celled, simple organism in the lab with all of our technology, and all of our experts have never been able to create life from scratch, despite what a lot of people think. There was a poll, a small survey, 2/3 of people thought that scientists have been able to create something as simple as bacteria in the laboratory. Absolutely not true. Even 1/3 of these people thought that scientists have been able to create things as complicated as frogs. Yeah, if you breathe it, if that ozone is not high in the atmosphere, ozone is one of the components they look at in smog when they tell you that don't go outside and exercise. It makes your lungs burn from the, yeah, it's a highly reactive oxidizer, yep. So let's look at a few other examples and then have a discussion. I'm not going to show you the whole pathway, but in the steps needed to make adenosine triphosphate, it requires 6 ATP. just to make one ATP. Nicotinamide adenine dinucleotide is an important cofactor in several enzymes. It takes NAD plus to make NAD plus. It takes tetrahydrofolate, which is why we need to have folic acid in our diets. But in the bacteria that make folate, or even plants, THF is needed to make THF. Circular causality. Coenzyme A is needed as one of the enzyme cofactors in the synthesis of coenzyme A. So if you're interested, Dr. Ann Gager, a research fellow at the Discovery Institute, she has a whole video up there, causal circularity in biology, where she goes through. I'll just show you some of the pathways when I scan through here.
To start the reaction.
So what do you do when here's a final product in this biosynthetic pathway is needed as part of the synthesis of itself? Like the THF would be needed right here by this enzyme in order to make THF. So it's got to come from somewhere else, which comes from somewhere else, and it's like, where did that begin? Which came first, the chicken or the egg? So she goes through and gives some of these pathways. You can see here, like here's THF, which is needed to make, it's not shown here, but at the very end, here you can see it more clearly. Here's the end product, tetrahydrofolate, but it's needed as a cofactor for this enzyme to convert right here. So what's not shown here, this is the product, this is the reactant, and there's an enzyme that requires ATP here, another one here. So what's not shown is the uber complex enzymes in between, where every one of these arrows is a complex enzyme. Some of which are regulated, especially the ones at the beginning or where there's branches and pathways. But yeah, isn't that interesting? It's needed here and it's needed here for its own synthesis. There's FAD. She talks about how you need FAD to make FAD, flavin adenine dinucleotide. All right, so what other examples can we think of? Well, think at the larger level, not just at the enzyme level, but what came first, the blood or the heart to pump the blood? Or the valves, like in the veins, that keep gravity from pulling the blood back down, so you assist getting that blood back to the heart, the deoxygenated blood. Or pressure sensors that say, hey, your blood pressure is too high or too low. Now we have to have these circular muscles in the vessels to constrict or open up, or the tissues to swell or stop swelling to control blood pressure. If the blood pressure gets too low, you go into hypovolemic shock. Or you try to stand up and you faint. If it's too high, look how many people are dying of hypertension because of, and you can even have like a extremely bad hypertension that can cause strokes and blood vessel bursts, aortic aneurysms. It can be deadly very quickly. What about clotting factors? I wish Dr. Boschkovich was here today because he specializes in the whole blood clotting cascade. And there's dozens of proteins, I think around 40 proteins involved in that. So imagine the first organism with a circulatory system. How does it reach reproductive age? How many of you have ever reached reproductive age without ever scraping your knee or getting a paper cut or any kind of cut that would cause you to just leak out and die? But what if you evolved the ability to clot, but not unclot or remove those clots later? Thrombolytic substances that break up that clot. Your whole circulatory system would turn into thick pudding and no more circulation. And some would say, is that unnecessarily complex? The cartoonist Rube Goldberg is like, okay, if all you wanted to do is start the toaster or whatever, do you need to have all these parts in between? But in the cell, those aren't happenstance intermediates. They sometimes serve other purposes. Sometimes they're checkpoints. Sometimes they're by being regulated. But they're all essential. It wasn't like the God or the Creator was just hey, let me show off, and he does show off. But these are all engineered to have a specific function. It's necessarily complex. So we talked about the enzymes that take the building blocks of DNA and synthesize them into long chains called DNA polymerases. There's also RNA polymerases. But you also have ligases, which can take broken DNA or cut DNA and glue them back together. And then you have nucleases. Some of them are like endonucleases. They start on the ends and just chop them all up into pieces so you can reuse them or break it down further. Some of them cut specific sequences. Bacteria have sort of a simple immune system by recognizing viral DNA that's inserted in them, and then they have an enzyme that will say, okay, I'm going to look for AAGCCT, and whenever I find that, I'm going to cut it. And they protect their own DNA by putting little methyl groups on there or tagging the DNA with chemical things that say, hey, this is you. And if it doesn't have that, it's foreign DNA and it's bad, it's probably a virus infection, chop it up. So there's dozens and dozens of these nucleases. There's whole companies based on using them as genetic engineering tools to be able to cut out like a gene and then insert it somewhere else using nucleases and ligases to cut and glue fragments of DNA together. So again, what happens if you evolve a nuclease that just chops up everything, goes unregulated? I mean, these things work fast. It would just turn your DNA into little fragments in a matter of minutes. All right, it's your turn. What other chicken and egg scenarios can you think of that have to do with, you think on the large scale too, just like whole global cycles like carbon cycles or photosynthesis or the need for oxygen? Yeah, two nitrogen atoms are triple bonded to each other to make nitrogen gas, and that's what most of our atmosphere is. At least 78% of the Earth's atmosphere is nitrogen. It's very difficult to break those because it's a triple bond that has to be broken. Lightning can do it. You get a little bit of this usable nitrogen as fertilizer through lightning, but lightning can also zap dead organisms. It will quickly destroy molecules and living things. So we have bacteria, like these purple bacteria that live inside of root nodules of legumes. They can take the nitrogen from the air that dissipates through the top layer of soil and fix it into reduced nitrogen or oxidized nitrogen. You can have like nitrates, nitrites. There's even production of ammonia, usable forms for life, for the plant fertilizer. And we need nitrogen to make amino acids. So yeah, everything seems to be interconnected. We've got 10 times as many microbes, bacteria, as we do our own cells on our bodies, in and on our bodies. And we would be dead without them. You try to grow up mice in a totally sterile environment, give them antibiotics and keep them in a germ-free cage, and they get autoimmunity. You have to fortify their food with all the B vitamins that the bacteria would normally make. You take them out of that environment, they die really quickly. They've had no immune training. It even takes certain germs to train your immune system to keep them from attacking yourself. So yeah, these germ-free mice will often develop autoimmunity, allergies, often lethally.
They look really bad too.
Yeah. So yeah, they're just the sheer number of interdependencies there are, and it's a far cry from Darwin's idea of survival of the fittest, where everything's trying to outcompete the weaker. In God's kingdom, it seems like there's this harmony, there's this mutual benefit. Now, when things go wrong, when you acquire mutations where you're no longer able to provide a food source from benign things like eating plants, then you have to become a parasite and leech off of some other organism like malaria does. They're like, oh, I can't make enough NADPH anymore, so I'm going to have to take it from the red blood cells and become an obligate parasite. So we see that degeneration. That's not part of God's plan, obviously. That's a whole other conversation of why are there so many parasites and seemingly natural evil in the world. Was that part of God's plan, or is this just a result of degeneration or maybe even malevolent design? chaperones. You guys know what protein chaperones are? It's these barrel-shaped proteins with a cap on them. And when a newly synthesized protein is made, often it's not folded correctly. It needs to have the right positive and negative charges aligned with each other. And there might be, 6, 7 positives and 6, 7 negatives, and they have to bind to the right pair. Or 2 sulfurs on cysteines have to line up and attach, but not these ones. So these nascent or newly built proteins that are synthesized by ribosomes go into these barrel-shaped chaperones. Sometimes the protein is even pre-coated with chaperonins to keep them from prematurely, sticking to the wrong amino acid pair. Or you want these hydrophobic regions to sort of form a cluster so that it'll coat them and prevent premature folding. It'll go in these chaperonins. Some incredible magic happens. No one really knows, because how can you get there to even look? And the lid shuts. Some things happen. Lid opens up. The protein comes out all folded how it's supposed to be. If it's not, there are recognition systems that says, you're the wrong protein. We're gonna tag you with this protein called ubiquitin and send you to the recycling bin, and you can, we'll start over, try to make another one. So that's a whole other recognition system. How do these proteins recognize when another protein is not folded right and decide whether to recycle it or keep it? And sometimes proteins are made right, but then get switched over time, like the L amino acid gets switched to a D amino acid. So you have a racemase that will flip it back where it's supposed to be. The left-handed is the natural form of amino acid. If it becomes a right-handed, it needs to be flipped back, or it ruins the function of that protein. So yeah, the problem is the chaperones are made out of proteins. So which came first, the protein or the protein that helps make the protein? Without those chaperones, a lot of proteins would just never get synthesized properly. Topoisomerases, that's a really cool. These are enzymes oftentimes DNA will get tangled up because you've got, like 6 feet of DNA crammed into the microscopic nucleus of every one of your cells. And during replication, the strands get pulled apart, but just like, the old telephone cords, when they start to get coiled around, you gotta sometimes it's easier to just unplug one of them and let them kind of dangle and unravel that tension and then plug it back into the phone. Well, there's enzymes called topoisomerases. One of them has these 2 hands that will grab onto the strands of DNA, cut one of them while still holding onto it, feed it through that cut so that it releases some of the tension, and then another enzyme called a ligase will glue it back together. So it keeps cutting, uncoiling the DNA and reattaching it. So what if it cut it, but it didn't have the ability to reattach it? And then you got all those unwinding DNA fragments. Instead of having, 23 chromosomes from mom and 23 from dad, now you've got 10 million really tiny chromosomes, little fragments or pieces of it. Probably 1%, less than 1%, and that's only of the stuff that we kind of have in common with organisms that are easier to understand. Some of these pathways you'll see in bacteria, others you have to study in mice, others you have to study in humans. It can take someone their entire lifetime and a full PhD career just to elucidate a portion of one of these pathways. We need computers to keep track of it all, because no one human, even if they had a photographic memory and an IQ of 300, could even store all of the information there is to know about biochemical pathways. Yeah, not just simple design either, but just an unbelievable genius level to pre-plan all of this and to think ahead of time, well, if I do this is going to cause this situation, so I need to have another system to take care of that problem to keep everything running. And so the amount of foresight, and when you look at these pathways and you see where they're regulated, think of an assembly line where you have all these textile resources and they have to be made into a final product, and there's a whole bunch of machines in between. Like, okay, this one's going to be in charge of sewing the button on, or this one's going to be putting a rivet on, or whatever. An engineer would say, well, when we need to stop this, because we've got enough of our finished product, where are we going to stop it? Are we going to stop this machine and turn the power off there, and then everything backs up behind it? Are we going to control the speed of the conveyor belts? Remember that I Love Lucy episode where they're trying to sort the chocolates and then it's going by too fast and they're trying to eat them and hide them, and because it's just, you get this backup of the supply chain. So where an engineer would put the on and off switches are exactly where you would expect them, at the right before branch points where you have two product lines splitting off, or right at the beginning, that's the regulated one. Sometimes, actually quite often, your finished product, as it starts to pile up, remember in a cell these can move around, it'll find its way back to the first enzyme and block that enzyme. So the more product you make, the less active the first enzyme is. And then as you start to run out of product because it's being used by the cell, now the concentration's going down, that's going to make that first enzyme start working again. It's called feedback regulation. Yeah, one estimate is there's like 10 to the 77 non-functional proteins. If you look at all the sequences for like 150 amino acid for every one functional protein.
Which is like all the atoms in the universe. Right,
Yeah.
Right.
There's also
And then there's
frame shift ones where like
yeah.
if you start reading at the beginning of a paragraph and it makes sense, every word makes sense in context, but then you shift all the letters over by one, all this, every space, every letter gets shifted over, and it makes another paragraph with another meaning. We might have time to talk about this later, but one huge problem is the fragility of these molecules, these biomolecules. Partly by design, you need the RNA to break down quickly because that's one way of regulating how much protein you're going to be translating from that messenger RNA. But the building blocks themselves, the cytosine is the most fragile of the base pairs, the bases, the nucleobases, and these things aren't going to just sit around in a fragile modified soap bubble, for millions of years until they get all the parts that they need to function, to even replicate. Yep. What came first, the mechanism of the eye, the muscles that control the eye, that control focus, or the part of the brain in the back that interprets those signals and says, hey, you're seeing red, or you're seeing a cat, you're seeing a flower, you're seeing food, you're seeing poison.
Right.
Yeah.
Yeah, which can, yeah.
Got to have the parts match up, or that's right. It's like, how do you go from asexual reproduction to sexual?
And just reproduction in general.
Yeah, just getting a cell to faithfully copy all of its. Right. Think about when a simple bacteria, I shouldn't even say simple because they're really complex. One of my professors is a biochemist. He specializes in E. coli, and he had 2 encyclopedic volumes about, they're probably this thick each, just on E. coli and how it works for what we know so far. We're still trying to figure out some stuff about E. coli, but when that bacteria needs to divide, think of the synchronization of events that has to happen. If it pinches in half before all the DNA is copied, this one gets 70% of the genome, this one gets 30, dead. Actually, we do. We have, in our gut, they provide all sorts of vitamins for us. It's the pathogenic strains. So sometimes they acquire these circular pieces of DNA called plasmids that will code for a toxin. And then good E. coli becomes bad E. coli. So it's like, you know, giving a gun to a person and making them crazy. You know, like they start shooting away the competition. And often they will have like bacteria like to have the nice, warm, nutrient-rich environment of the gut to themselves. So they will actively produce antibiotics that will kill off the competition, while also on a separate plasmid producing the enzyme that chops up that antibiotic so it doesn't kill itself. So what if it made the antibiotic first, poisoned itself? Oops, guess I didn't get to pass on that gene, unless you had the antidote to the poison, also made at the same time.